Okay, it is the genetic series. I welcome you to module 10. And in this module, we'll be looking at variation and components of variance. So, what is variation? You'll notice that when we look at different animal species, there are phenotypic differences between individuals within a breed and even within families. For example, if we are to take a look at cattle, we see obvious differences in their coat color. We see differences in their horns. Some have horns, some do not. Some are very heavy, some are not that heavy. The quantity of milk produced, for example, in some is quite large, and that may not be the same situation in others. Now, these differences that we notice in individuals, even though they are of the same species or breed, for certain traits or character is what is referred to as variation or variance. This variance can be classified into two or more categories, and this categorization depending on the character being examined and also on the unit of measurement that is being used. So those characters that are classified into two or only relatively distinct categories are described or are said to be showing discrete variation. But those that could be categorized into a larger number of categories are limited to units and accuracy of measurement used for classification are said to show continuous variation. Now, variation in size, in the rate of growth, for example, efficiency of feed utilization, carcass characteristic, disease resistance have been observed in animals. And this variation to occur, both the genotype and the environment are important in determining the observed differences among individual animals. In some instances, there are specific hereditary and environmental influences that may produce variability, which is or may not be directly attributed to either the gene or the environment alone, but is as a result of an interaction between the gene and the environment. Majority of characters in animal breeding are continuously changing in the sense that individuals cannot be easily classified into distinct classes. So continuously varying characters are usually known as quantitative characters or metric characters, and examples are as shown: milk production, fleece weight, body weight, egg number, body length, and many others. So the measurement of the variation in a trait or a character is called variance, and variance can be divided into different components. So what are the causes of variation in animals? All the traits in animals are classified to be either qualitative or quantitative. Qualitative trait falls into few clearly defined classes, and this is because they are usually influenced by one or very few pairs of genes, and how they are expressed may not necessarily be greatly influenced by external environmental factors. The polled or horn consideration in cattle is an example. Some cattle have horns and some don't, and this having or not having is controlled by one or very few pairs of genes. That's what we are saying. On the other hand, variation from small to large as is found in milk production, or the rate of gain, examples of quantitative variation. Different cows will produce different amount of milk based on their genetic makeup. For example, the Holstein Friesian is not expected to produce the same amount of milk when you compare it with the Jersey or the Cementa or even the white Fulani cattle that is found in most parts of Africa. Likewise, their rate of gain are different. These traits are examples of quantitative variation or quantitative traits. So the study of variation involves two basic stages or phases. One, or firstly, is to describe the phenotypic variation, and the secondly is to translate these phenotypes into genetic terms, and then redescription of the variation genetically. So variation among phenotypes in a population will arise from three different sources. First is the average differences due to the genotype. Second is variation from the environment. And thirdly, interaction between the genotype and the environment. So if we are to define phenotypic variation, we'll say it is the observable or measurable differences among individuals within a population for a particular trait of interest. Let's look at genetic sources of variation. Genetic variation is a measure of the amount of heterozygosity at a locus in a population, which is usually given by the total frequency of heterozygotes at a gene locus. One important thing to note is that the genotype of an individual is fixed at conception, and except there are extraneous situations like mutation, it is expected to remain the same for the remainder of his life. His genetic makeup is actually determined by the genes that is transferred from both parents. So the individual as well as his parent possess thousands of such genes, and it is expected that members of an inbred animal family should look more alike genetically than those that are not inbred. The degree of genetic similarity is expected to increase with increase in inbreeding, especially if selection is done directionally. So parents that are homozygous for many pairs of genes, we have offsprings that are more alike genetically than parents that are heterozygous for several genes. And the degree of homozygosity actually limits or decreases genetic diversity in populations. In fact, genetic variability within a species is almost unlimited. The second source of variation is what is known as environmental source of variation. And phenotypic variations in economic traits, for example, could also be due to environmental influences. So environmental sources of variation are also very important. And environmental sources of variation includes things like disease, nutrient, temperature management, accidents, and others. You notice that these are factors that are external to the animal itself. And as animals encounter these from time to time, it changes its productivity. So phenotypic variation due to the environment are important. The reasons are that one, for A, they are not transmitted from parents to their offspring. So the environment needs to be well taken care of. Because B, they can overshadow whatever variation that is due to the genetic makeup or that has been transferred from the parents to the offspring. And C, The proper environment is therefore necessary for an individual to reach its full genetic potential. Finally, D. Rapid improvement can also be achieved in livestock production when we supply uniform or superior environmental conditions to breed animals. So what are the components of variance? We had said that quantitative traits are controlled by genes at many loci and also by environmental factors such as nutrition, climate, disease. So classifying animals based on individual locus may not be an easy task as the loci are not easily distinguishable. So when we want to quantify the productivity of animals, we simply do that by measuring what we're interested in, and this could be in kilogram per day or growth rate, for example, or in the number of eggs that are produced. So this is a measure of their phenotype, and we therefore call this their phenotypic value. Phenotypic variation is the measure of observed differences, therefore, between animals within a population for a particular trait. The phenotype and the genotype are therefore considered as raw materials when we want to improve animals. Because selection is made on the basis of how the phenotype and the genotype varies. Phenotypic variation observed or measured in animals is due to the effect of the gene and the environment. And this is presented by this expression: phenotypic variation is equals to variance due to the gene or genotype plus variance due to the environment. So this is the definition of the parameters. Variance due to the environment includes the effect of nuclear genes as well as mitochondrial genes and interactions between genes. So we want to look at the genotype. The genotype of an animal is made up of a large portion due to genes with additive effect. This is presented as VA. Now, this is the variance in a trait that is due to the adding up of all the effects of all the individual alleles without any interaction with other alleles. And it actually represents the average inherited effects from the parents. Hence, variance due to additive effect is transferable directly to the offspring. And this is known or referred to as the breeding value of the animal. There is also dominance effect of genes represented by VD. Dominance effect of genes is the variance due to interactions between alleles of the same locus. This variation at a locus is due to the dominance of one allele over the other, and this is not directly transferable from parents onto their offspring. And why is this? Is because the offspring receive only one allele or a gene component from each parent, and the possibility of receiving, say, an A as against a small a is purely due to. The third aspect or component of the genotype is due to epistasis and other interactions presented by VI. Now, this is due to interactions between non-allelic genes, and therefore we can express the variance due to the genotype as VA, that's the additive genetic variance, variance due to dominance plus variance due to epistasis and other interaction. Now, the environmental variance is also very important and actually consists of permanent factors that are inherent or inside of the animal, and this is presented by EP. And this permanent factor is the environmental effect that is unique to each animal, which permanently affects the expression of a repeated trait. The second aspect is differences, temporary differences in the environment that are external to the animal, represented by ET. And this is defined as the environmental effect that influences a simple performance record of an animal, but which does not permanently influence the animal's performance potential for the repeated trait. So these changes, temporal environmental differences, cannot be controlled uh, by the animal. Likewise, the permanent environmental influences. So the environmental variance can now be written as being made up of permanent environmental differences as well as the temporary environmental uh, influences. Therefore, the phenotypic variance can be said to consist of variance due to additive effect plus variance due to dominance plus variance due to epistasis and other interaction plus permanent environmental variance plus temporary environmental variance. But among all of these variances, the variance of the breeding value, which is a component that is passed on to the progeny, is very important in animal breeding. So it is important to estimate the proportion of the total variance that is caused by both genetic and environmental effects, as it is vital in the improvement of phenotypic traits. If the variation that is due to genetic effect is high, selection of individuals will most likely lead to improvement of the trait of interest. And if the environmental effect is high, and by implication this means that the genetic effect is low, then other approaches like crossbreeding will be better employed to bring about improvement in subsequent generations. So it must be noted that estimates of genetic and environmental variance are specific to a population and to a particular set of environment where the estimates are made. Separation of variance into genetic and environmental components does not separate their causes of variation. Therefore, such estimates for a trait cannot be generalized. So we can obtain sample and population phenotypic variance for a trait using the formulas given below. So this is sample variance, and this is the formula, sum of xi minus xi bar squared divided by n minus 1. And for population variance, the same thing, but this is divided by capital N. And this is the definition of the parameters. So we want to carry out a work example to see how we can apply the formula. We want to consider data that will be on the next slide of average egg weight of Fulani ecotype chicken sampled in our environment. So we want to estimate the phenotypic variance for egg weight of the Fulani ecotype chickens. We want to also estimate the standard deviation of the measurement as well as the standard error of the trait under consideration. So in this slide, what we have is the egg weight. This egg weight is collected from 10 different beds. And so these are the egg weights. So when we sum up, we have 258. This is the mean, and that is 35.8. So in this column, what we have is xi, this is xi, minus xi bar, meaning each value here, from each of the values here, we're going to subtract the mean value. And if we do that, these are the values you get. In this column, what we have done is to square each of the values here. And when we sum up each of the squared values, we have a total of 625.6. Now, from this, we can now go ahead and get the variance, the sample variance using the formula, and this is what we get. We can also estimate the standard deviation, which is the square root of the variance. So the phenotypic standard deviation is square root of 69.5.1.34. And then we can also estimate the standard error using the phenotypic standard deviation divided by square root of n. The answer is 2.64. Covariance. Now, many of the genes on the chromosomes have effect on more than one trait or character at the same time. This condition is known as pleiotropy, and the effect is known as pleiotrophic effect, which is that the ability of a gene to have control over the expression of more than one trait at the same time. Okay, so a change in a gene that is pleiotrophic will affect all the traits that it controls, or all the traits that are influenced by that gene. So such traits that can be influenced by the same gene or groups of genes are said to be correlated. This is because a change in one of the traits will invariably lead to a corresponding change in the others too. And these changes in traits that are correlated is measured by using their covariance. So the total phenotypic covariance represented by this between two traits can be partitioned into a genetic and an environmental component. Genetic covariance estimates covariance in two or more traits due to a common gene effect, while the environmental one looks at the covariance due to also a common environmental effect. A knowledge of both genetic covariance and environmental covariance that exists between traits is vital in developing selection procedures for different traits in livestock. So let's assume that we measure two traits, body weight as well as body weight, represented by X and Y respectively, and the sample covariance between the two is denoted by x and small y, and the values for trait X and Y is denoted as below. So these are the values for trait X, and these are the values for trait Y. So using this, the process for calculating sample covariance is similar to that of variance, sample variance. So the sample covariance between X and Y is given by this formula, where X, mu X bar plus mu Y bar means the sample means for body weight and body weight respectively. So this is the core population covariance, and the parameters have been defined. So when the covariance is zero, it is suggesting that the traits under consideration are totally independent of each other. This means that there's no common gene effect on the two traits concerned. When it is positive, it means that there's a positive common gene effect on the two traits. The implication here is that as one of the traits is increasing in magnitude, the other trait is also expected to increase in magnitude. There is also the possibility of a negative covariance, which means that as the value of a trait is increasing, the corresponding value of the other trait will be decreasing. In this case, we say that there's a negative common gene effect on the two traits concerned. And such a negative covariance is only expected in a situation where the genes that improve a trait tends to have a negative impact on other traits. A typical example is egg weight and egg number. You would have been noticed that as the egg weight increases, there's a corresponding reduction in the number of eggs, and the reverse is also true. Genotype and environment interaction. The interaction between the gene and the environment means that animals of a certain genotype may perform more satisfactorily in some environments than in others. In other words, while one environment may permit the expression of the genetic characters or potentials in an animal, another environment may not. So we say that a genetic stroke, times environment interaction exists when the ranking order of individuals or breeds change in different environments. If, for example, on a pasture, animals are evaluated and ranked as given here. This is a ranking for 7 different bulls. Okay. Now, when these same animals were changed to concentrate diet and they were evaluated for a trait, the ranking changed to something like this. Then, in this kind of situation where there's an almost complete reversal, we will say that it would be unwise, for example, to select the bulls than in the environments in which they will be used. So if the animals are going to be on pasture, it would be wise to use this ranking for selection. And if they are going to be on concentrate diet, it would be wise to use this ranking for selection. That's the implication. In other words, if the results of comparison vary based on the environment in which the trials are conducted, we say there's a gene and environment interaction. This variation results may involve just a small change in the relative performance or a change in ranking. And the problem with gene and environment interaction is that it is difficult to predict in advance whether or not such an interaction is likely to occur, or whether such an interaction will be important in evaluating the performance of the animal. This is because there are many environments and there are many genotypes. So it is impossible to say that genotype and environment interaction will not occur in a species. In fact, it is very important for the farmer to be aware of this so as not to expect fantastic results from animals that were evaluated in a different environment. Environmental evidence suggests, for example, that dairy and beef cattle have fewer important gene and environment interactions, whereas these kind of interactions are very important in sheep and in pigs. So if gene and environment interactions are important for a trait, then it is usually better to select for that trait in an environment where the livestock is supposed to be used. What this means is that we cannot select animals in Europe, for example, and then bring them down to Africa and expect them to perform similarly. The reason is that these animals were evaluated in Europe, and if they perform very well, you bring them down to Africa where there's a different set of environmental effect, the animals may not likely perform very well because of the gene and environment interaction. So if animals are to be used in widely different environments and different management regimes, there's a great responsibility on breeders to be aware of these possibilities. So where there is gene and environment interaction, the phenotypic value of the individual is not simply just phenotype is equals to genotype plus environment, but in this case, it's going to include also an interaction effect or component, which is the gene and environment interaction. So the phenotype of the animal is now made up of the genotype plus the environmental effect as well as the effect due to interaction between the genotype and the environment. The interaction component is now will give rise to an additional source of variation, and so the phenotypic variation will be due to variation due to additive uh, or the genotype represented by the additive genetic variance plus variance due to environment plus variance due to the covariance between the genotype and the environment, as well as variance due to genotype and environment interaction. So genotype and environment interaction becomes very important if animals of a particular population are to be reared under different conditions. For example, if a breed of livestock will be used by different farmers who treat it differently, the different treatments have introduced a different environmental influence, and this may affect how the animal genetic component will be expressed. So, under such differing conditions, animals should not be expected to perform in the same way. So, we have come to the end of this module and we have looked at variance and the components of variance. In the next module, we'll be looking at how to measure heritability as well as repeatability, and we'll also look at their importance in animal breeding and genetics. We'll see you there.